All right, everybody, let's go ahead and get started for today. I don't know if anybody has seen yet, but I sent an announcement this morning saying that my office hours have changed. They're going to be from 11 to 12 today. So I didn't tell anybody, but I am getting married this weekend. So I've been kind of swamped with a lot of wedding stuff. So the earlier I can leave, the better. So that also means I'm probably not going to be responding to any emails this weekend. I probably won't get to them until Monday, Tuesday at the latest, depending on how many people email me over the weekend. So if you guys have questions, come during my office hours right after class, because it'll save me a lot of time. Instead of having to write to everybody, I can just answer your question out loud, and it'll save me a lot of time. Oh, and thank you everybody for all the congrats. So let's see, let's go ahead and get started. So today we are going to be talking about viruses. So as I talked about a couple of days ago when I was introducing the syllabus, I feel like with how coronavirus has changed our society, I think it'd be a good idea to understand how this works. But before I get into the details of the virus, I kind of just want to introduce you to the world of viruses and what's out there. And so I don't really expect you guys to memorize like all the viruses that exist, but I will highlight some key ones that I feel that are important to know, because a lot of them that I will be touching on still affect us today, or there was recent outbreaks that you may or may not have heard about. So let's dive right into it. So the outline of today's class is going to be a brief history on the discovery of the viruses. And then we'll go ahead and talk a little bit about how we're actually able to visualize the viruses. I'll get into the general size and structure of a virion, and I will introduce how we classify viruses. And then I'm briefly going to talk about how viruses get into a cell, but that's going to be the main focus for the next lecture, when we're emphasizing how COVID gets into our cells and hijacks our machinery to replicate. And then I'm going to give you a little intro into the treatments and tools we have to fight against viruses. They're just going to be it's just a preface to tools that I'm going to end up discussing in more depth after we learn about immunology. But I think it's a good idea to kind of have a general sense of what these are and a general sense on how they work. So for today, what I really want you guys to know, and if you know these things, you'll do fine, because my questions are going to be all connected to the learning objectives. So know what makes up a virus. Understand that there's going to be three main shapes that exist for viruses. And you might want to know an example or two for each of these. Usually I'll probably end up asking a question about one that I give you an example of, so just know it. Understand this concept of sense and antisense when I'm referring to genetic strands. So I had mentioned in yesterday's class that when it comes to a DNA converting to RNA, one of those DNA strands is important for being used as that template strand to synthesize the RNA. And so today I'm going to get a little bit more in depth in all of that, because viruses come in a variety of shapes and sizes, and their genome composition is no different. And so when I refer to the different kinds of viruses that exist in terms of referring to their genetic composition, I would like you guys to understand those, because you will get questions on those. And then this will link to understanding the Baltimore classification system that is used to group the different viruses, because there's going to be two different naming systems, but I want you to focus on the Baltimore. And then know the tools that we have to fight against the viruses. So to start us all off, are viruses alive? I wanna hear people's opinions on this. And go ahead and respond, and I'll open this up until we have at least 80% of people that have responded. I always find these to be so interesting. All right, I'll close this in 5 more seconds. Majority of you guys have almost answered. 3, 2, 1. All right, and poll. So I'm going to share these results. So the majority of people say yes, viruses are alive. And I will go ahead and ask this question again as I go through the lecture, and then let's see what you guys believe in. And I'll go ahead and expand a little bit on this. All right, so they are, but aren't technically. Yeah, we'll cover some of that. All right, so as I mentioned, I'm never going to have you guys memorize people in history, but I do believe it is important to give credit where credit is due, especially to the people that pioneered the field of virology. And so here we have Dmitry Ivanovsky, and he is or was a Russian microbiologist. And he is one of 2 scientists that's credited for discovering viruses. And his work was all relevant to trying to understand what causes tobacco mosaic disease. And so this is a kind of infection that infects tobacco plants. And so we can see here an infected leaf. So he ended up performing some experiments where he ended up taking a filter. Well, basically, he broke up the leaf, an infected leaf, and then ground it all up, put it in solution, and then filtered basically all the stuff that was in solution, and then took some of that solution and then put it onto an uninfected leaf, and saw that that also got infected. And so he originally thought, because this filter is able to filter out bacteria, that it was a kind of toxin that bacteria was able, that's producing, that's causing this infection. And so it really wasn't until the electron microscope was invented in the 1930s that we're able to visualize what the tobacco mosaic virus was. And so here we have Martinus Beijerinck, and he was a Dutch microbiologist, and he was convinced that it wasn't a toxin, but this new biological agent. And so he ended up repeating Ivanovsky's experiments, but hypothesized that that there's something else. There's a virus that's infecting these plants. It wasn't a toxin. And so he coined this term, contagious living fluid, which at the time was used to describe viruses because we didn't really have a name for it. And so both him and Ivanovsky are credited for being the fathers of virology. And here we are today learning about all these different kinds of viruses that exist in our world. So what is a virus exactly? So a virus is a non-cellular particle, so it's not a cell, which is made up of genetic material that is encapsulated by a protein. And it invades living cells in order to live. And so because of that, it's considered this obligatory intracellular parasite. And so all this fancy word means is the virus needs a host cell to survive, just like a parasite, which it needs a host to live in. Viruses need to get into a cell and hijack the machinery so it can replicate itself. And then I will talk about more about some distinct ways that viruses can multiply when in the next lecture, when I get more in depth about the actual hijacking of our host cell machinery. Viruses do not have functional organelles, cytoplasm, or cytoplasmic membrane. So although they do have ribosomes to synthesize, actually, no, I lied. They do not have ribosomes. They use our material to hijack everything. So they don't have any kind of organelle to basically produce the RNA or DNA or whatever genetic material they have. They don't have any kind of jelly-like substance that's going to keep the inside suspended, and it doesn't have a membrane that regulates things that go in and out of the virus. And so this also means that it does not have any metabolomic activity. And so I'll talk more when we're going into the different molecules of life, but like sugars, fats, and proteins, they can get broken down to be used for energy, just like when we eat stuff and then we're able to do stuff. Well, viruses don't do any of this. They don't have mitochondria to utilize sugar to make ATP, or they don't have any kind of lipid transporters or scavengers to do the same. So they have no metabolomic activity. So why do we even care about studying viruses? Well, viruses are an important disease-causing agent, and they infect all forms of life. So it's not just humans and animals. They infect plants, like I showed you with the tobacco mosaic virus, and they also attack bacteria. And I will talk more in next week's lecture about bacteriophages and what they do when it comes to infecting a bacteria. But here we have an image of a bacteria getting infected by bacteriophages. And so we also care about studying viruses because we know that they transfer genes into organisms. And scientists have exploited this to use tools for biomedical research to understand how genes function and their implications in disease. And with that knowledge, we've been able to translate that into patient care, where we're now able to basically engineer viruses to carry a specific sequence that we want them to carry, so they can infect cells and transmit any of the information that we want present into the cell. And so I'll highlight this when we're talking about vaccines in a couple of weeks, because the Johnson & Johnson vaccine uses adenoviruses to basically transport the sequence that it wants, so the host cells can express the proteins of interest. So how big are viruses? They're actually very, very small. So typically the size of the virus is 100 to 1,000 times smaller than the cell that they infect. And they're much smaller than a bacteria. And so we have a nice diagram here of showing the size of a typical bacteria and a series of different viruses and their relative relation to the bacteria. So here we have a couple of the quote unquote giant viruses. And then, but typically most of the viruses are probably within like the 300 or smaller nanometer range. And viruses can only be seen with an electron microscope. So unlike fungus or bacteria, where if there's enough of them in a space, we're able to actually visualize them. If there's a lot of virus in one place, we're still not going to be able to see it because of how small they are. And we're able to see them now because of this technique called electron microscopy. And basically, there's two ways of being able to visualize the virus. So here I have an image of the scanning electron microscopy of coronavirus highlighted in magenta. And then here we have a transmission electron micrograph of the coronavirus highlighted in orange. And so what's really the difference between the two? Yes, they are both types of electron microscopy. However, scanning electron microscopy, or SEM, gives you the topography of the object, meaning it's going to look at the outside of the object of interest. So think of it as taking a picture of the outside of this apple. SEM images are only going to be able to visualize the outside of the object. Compared to transmission electron microscopy, it's actually able to penetrate deeper into the object. And so what it does, it actually gives you a cross section or a snapshot of one of the layers of the object of interest. So you can compare this to a cut apple. Basically, it can look at a specific layer and look to see what is going on inside it. And so what I wanted to take 5 minutes to do is go ahead and have you guys watch this video on how this virus this technology works for the virus, because I feel like this video does a better explanation and simplification of how it works more than I could. And you know, this is relevant to coronavirus, and I feel like there's a couple of cool things to learn in here.
This is it.
Hopefully you guys can all
This
hear.
is
If you can, just let
SARS-CoV-2,
me know in the chat.
the novel coronavirus that first appeared in humans in late 2019. These are some of the first close-up views of the virus, made using a very specific imaging technique that can see things far too small to be visible under a normal microscope. They show us how the virus moves inside the human body and how it hijacks our cells using these. These spiky crowns give the coronavirus its name, and they're the key to understanding how to beat it. To get this right, I needed to bring in two experts. The first is my colleague Liz's dad, Frank. He teaches material science and engineering at Ohio State University.
Liz tells me that you are interested in electron microscopy.
Yes, so that's how you pronounce it. Okay, that's a good start. And the other is Beth Fisher. Her team at NIAID, the National Institute of Allergies and Infectious Diseases, created those initial images of the virus, the ones you'll start to notice embedded in news articles around the internet.
I think for a lot of people, there's a lot of mystery to this virus as there are to other viruses that emerge.
So let's start with how these images were made. The first thing you need to know is that the coronavirus is very, very, very small, around 100 nanometers. To put that into context, if you take out a ruler and look at one of the millimeter markings, you can fit 10,000 virus particles inside of that. At that size, it's invisible to us, even under a standard light microscope like the one you might have used in grade school. That's because the smallest wavelengths of light humans can see measure around 400 nanometers, not small enough for the coronavirus to be visible. In order to see something that small, you need an electron microscope. How does it actually differ from what we would think of as a microscope?
Instead of light, we're using electrons, and electrons you think of as particles. But if you take the electron, strip it off of the atom, and accelerate it in the field and make it fly really fast, if you make it move fast enough, it actually will behave as a wave as well.
And that wavelength is much, much smaller than the light waves we use in a standard microscope.
So now you're like 6, 7, 8, 9 orders of magnitude smaller in size. So now you can see smaller stuff.
Look through NIAID's coronavirus Flickr page, you'll come across two distinct types of images, SEM and TEM. They're made using two different types of electron microscopes. A scanning electron microscope, or SEM, scans the surface of a sample and records what bounces back, sort of like how satellite imaging works. It gives you the basic topography of whatever you're looking at, with realistic lighting similar to photography. Shadow and relative size of objects shows you their placement and how they move through the cell. A transmission electron microscope, or TEM, goes way deeper. It records the inner details of a sample by transmitting electrons through it and projecting a cross-section of its inner structure.
So it's part of the basic science research to try and understand structurally what's going on.
Images from both microscopes are taken in black and white. The color is added later for clarity, like in this SEM image, where virus particles, colored in yellow, are seen emerging from the surface of a cell, colored blue and pink. When examined together, these images can help scientists start to figure out how the coronavirus works.
There's ways to start then understanding how is it getting in a cell? How is it harnessing that cell machinery to make more of itself?
Can you just tell me kind of what we're looking at here?
So this is a single viral particle, and the yellow you see is the core of the virus itself, and then the corona, where coronaviruses get their name, is sort of that halo around it that's in orange.
That corona is the key to understanding how the virus hijacks our cells. The spike proteins that surround the virus attach to a host cell's membrane and then penetrate it. Once it forces its way in, it spreads its RNA around the host cell, multiplies, exits, and repeats, which makes us sick.
So if we can bind those spike proteins up with something like an antibody, it'll prevent them from being able to attach and enter cells.
Which is exactly how we've beaten back viruses with similar spiky proteins before.
This was a 3D rendering of the Ebola virus. You see all those proteins on the surface. So when we talk about the spike proteins, that's what we're talking about, no matter which virus we're looking at. This is HIV, actually. So this is printed after we do our cryo-TEM, and you can see all these little tiny proteins distributed on the surface. And these are the proteins that we tend to target for vaccine development. You want to lock these up so they're tied up and unable then to stick to a cell surface.
Is there anything about the images specifically that you'd want to share with our audience that maybe you think would be helpful for them to know?
I think when you can face your enemy, it takes a little bit of the fear factor out of it. I think it's just understanding what it is we're looking at, how it works within our bodies, but to show that this is something that can be tackled and overcome, I think it's important to know.
All right. So hopefully you guys found that video useful and interesting. So I'm not gonna like ask you guys to remember like all those steps to how this type of microscopy works. But what I would want you guys to know is if I were to give you an image, either an SEM or a TEM image, I'd want you to be able to identify what you're looking at. So I feel like that video and this slide will help you be able to decipher. So viruses are very diverse. They come in a variety of shapes and sizes. You do not have to know all of these, but the ones that I highlight in this lecture, I probably will end up asking you. So I'm not going to ask you anything unfair. Most of the viruses that I do cover are probably ones that you have heard or you should have heard. But yes, they come from, almost 1,000 nanometers all the way to 20 nanometers. So viruses have three main shapes. So it's really dependent on what text you use and your source of information. But for simplicity, somebody is asking a question or raising their hand. Feel free to unmute yourself and ask.
Hello, Professor, can you hear me?
Yeah.
Hey, I had a quick question regarding the Vox video about COVID.
Yeah.
So I think at this point it would be safe to say that maybe COVID was probably modified inside of a lab. I'm curious like what your explanation of, like let's say a scientist did modify it and they say that they made it so that it's more easily transmissible from person to person. Do you understand how that process would work? Like if it were true?
I could get more. I'm going to get more into that next week. But simply what you'd want to do for that, and hopefully no one's going to take this information for bad, but you'd want to modify proteins that your body's never going to be able to recognize. So you'd want to. We're currently developing vaccines and whatnot to educate our immune system, and if your body has never seen a certain sequence called a peptide before, then it's going to have no immunity against it. And so I think if you really wanted to genetically modify a virus so it's more virulent and it can spread better, I would probably design particles, especially those spike ones that we're targeting now, to encode a sequence that your body has never seen. So you probably would want to figure out what is the current sequence for the spike proteins, and then compare that to see if there's any other organisms, including other coronaviruses, that have that sequence, and then modify it in some way that where it doesn't match any other organism. And if it doesn't match any organism, then you're not going to have any kind of immunity against it. Does that kind of answer your question? It's kind of
yeah, it's an interesting perspective on it. One thing I read that I thought was interesting was something with fungi particles that they would attach to the virus. Do you understand that, like what that would do specifically?
I don't, but I can look it up and I will let you see if I find any information on it. But yeah, I'm all for keeping the lectures interesting and keeping an open dialogue to some of these things that we still are learning about, because I feel like the previous professors that have been teaching this class, they started introducing virology concepts, but it's been developing this last year because we're now barely having vaccines. We're barely learning how they work, but a year ago we didn't have any of that. So yeah.
Right. All right, well, I appreciate it, Professor. Thank you.
Yeah, no problem. All right, so going back to the three main shapes. So we have a helical or filamentous shape to it. And so a prime example of a virus that has this shape is Ebola. And basically it takes on this worm-like structure. And all helical or filamentous viruses are going to have a similar shape. The other kind of shape are polyhedral viruses. And so with polyhedral viruses, it just means that it's going to have multi faces to it. And basically with these multi faces are going to have is going to describe this shape. However, there are some viruses, when you look at them, they have more of this globular or spherical shape to them. However, if you look at the proteins that are actually encoding or what's present on the surface, you actually see that there's multiple faces also present that might not have this, I don't know, very polyhedral shape. But when you look at it in depth, it still takes on this structure. And then the last viral shape are complex viruses. And basically, these are ones that aren't going to be these worm-like structures or have this circular or multi-faced structure, but might be a combination. And so a prime example, and I'm going to talk about this particular virus multiple times throughout this class, are bacteriophages. And so bacteriophages basically are the ones that you typically see in the movies when Hollywood's depicting how a virus goes and infects cells and stuff. Bacteriophages only infect bacteria. It doesn't infect humans or any other kind of organism. And so these are typically going to look kind of more scary, really. They have this head to it, and then this body, and then their basically their tails or protrusions that are going to be used to basically stick itself into the cell surface of a bacteria. And then this portion here ends up drilling a hole into the bacteria and depositing all of its genetic materials so it can hijack the host machinery and replicate. And I'll be getting more into that, that whole process when we cover prokaryotic viruses next week. So the structure of the virion comes into two main parts. There's the core, and then there's the protein coat. And so the core is basically the genetic information that encodes the virus. And this is typically going to be either a DNA or RNA strand or strands. And this is only going to encode a few hundred genes compared to the human genome, which I discussed yesterday, which encodes over 20,000. So these are very small genomes, and they don't really encode for a whole lot of proteins. Basically, the proteins they mainly encode for are to build its structure and to build proteins that are going to help hijack the host machinery. And then the other part is going to be this capsid. And so the capsid is kind of like our nucleus, but not because it's not membranous or as complex, but its purpose is to protect the genetic material of the virus. All viruses have a capsid. However, only some viruses have an envelope, and the coronavirus is an example of a virus that has an envelope. And basically, this is just another layer that protects the genetic material and also helps viruses hijack or get into the cell. And so this envelope is going to be consistent of two things. It's going to be it's going to be composed of lipids. So just like our lipid bilayer that we have on all of our cells, they're going to have the same structure. Is going to be a lot different. It's mainly going to be phospholipids, but that's all. There isn't going to be any kind of transporters or receptors present. And then it's going to have embedded in it some, they're called glycoproteins. So basically they're proteins that have sugar residues on them. And the whole point of these is to help bind to the cell so it can facilitate entry. And so the core and the protein coat make up the virion. And so people use virion and viruses interchangeably. Basically, a virion is the whole structure of the virus without it being inside a cell. And then once it's actually in the cell or in the process of budding off of the cell, people will call that a virus or productive virus. So when it comes to classifying viruses, people can't really agree on a lot of how to name them. So there's a couple of classifications that are used to kind of group them. And so we have this ICTV classification and this Baltimore classification. And so the ITC, or the International Committee of Taxonomy and Viruses, basically is responsible for creating their own version of classifying viruses. And they use the Linnaean hierarchy of systems. So, going through the kingdom and working your way all through species. And so when they're considering how to go about naming these viruses, the question they usually ask themselves is, what is the morphology of the virus? What is the size? What is the shape? What does it have an envelope? Those are all of the criteria that are considered when it comes to going about separating where they should be grouped. The other thing they consider is what's the size of the virus, what's the weight of the virus? And so the weight is really determined on the structural proteins that make up the virus. And so you're able to figure out what the weight is by looking at the amino acid sequences, and all the amino acids have a certain weight to them. So they can go ahead and estimate that. And then the last consideration when it comes to figuring out where these group together is, what does its genome look like? And basically, the genome is going to be composed of DNA or RNA, and then they consider, okay, is it double-stranded or single-stranded, and does it have a positive or negative sense strand? And again, I'm going to go into this in a couple of slides of what this is. And then with the events of sequencing, where we can actually isolate DNA or RNA from cells or viruses, and figure out what are the actual letters that make up the whatever it is that it's trying to create. And with the event of the sequencing technology, viruses have started to be grouped differently, just because there are some viruses that, even though they might not look the same at a genetic level, they are more similar. So then they're going to end up being grouped more closely together. The other classification, which I'm going to be focusing more on, and what I want you guys to actually know, because you will be getting questions on this, is the Baltimore classification. And so the reason why it's called the Baltimore classification is because of this guy, David Baltimore. And basically the way that this classification goes is it's all dependent on what mRNA mechanism is used by the virus to generate its proteins. And so all viruses, just like humans and all other organisms, they need messenger RNA to make protein. But depending on what kind of genetic material it has will determine on what kind of mechanism is needed to generate this mRNA strand. So you have to consider, is it DNA or RNA? Does it have one or two strands to it? And if it does have one strand, is it positive or a negative sense strand? And then finally, you have to consider if it uses this enzyme reverse transcriptase. And so all reverse transcriptase is an enzyme to make DNA. With this kind of classification, there are 7 different groups that are generated. And so to kind of talk a little bit more and expand on some of this classification, we need to understand what reverse transcriptase is. And so as I mentioned in, I forget what lecture, ACE just implies that it's an enzyme. So reverse transcriptase is an enzyme. And so remember how yesterday I went over this concept of transcription where DNA goes to RNA. Well, the reverse process of that is reverse transcription. And so that means that this enzyme, reverse transcriptase, needs RNA in order to make the DNA. So really understand that concept, because it's going to help you when it comes to doing some of the problems that I have in a couple of slides. But so reverse transcriptase needs RNA to make DNA, okay? Is there a question here? Is DNA just strands of the RNA? So not when it comes to viruses. So viruses, have their own unique set, and I will get into that in one more slide. So I want you guys to understand this concept of sense versus anti-sense. And this all comes down to nomenclature. And so going back to what we learned yesterday and the day before, DNA has two strands to it, right? We have 1 and 2. But as I was also talking about yesterday, only one of those strands gets used to make RNA. And I didn't want to get you guys confused with all this nomenclature, but I feel like this is the perfect time to introduce it because it's important to when it comes to understanding the nomenclature of viruses. So it's always going to be this 3' to 5' strand, which has a few different names. And so the names that I want you guys to know are anti-sense strand, template strand, and minus or negative sense strand. And I'm gonna use some of these interchangeably when I'm talking about these, but these are gonna be the ones that are really important to know because you need this negative strand to make mRNA. And so once you have this strand, you can go, transcription can occur to make mRNA. And so the other names for this strand that isn't used to make RNA is the 3' strand, sense strand, non-template strand, and positive sense strand. But again, for stressing purposes, it's going to be this, what's highlighted in red is what's going to be important to make the mRNA. So here is a beautiful diagram of the different groups of viruses using the Baltimore classification. So we have DNA, RNA viruses, and they're called retrotranscribing viruses, meaning they need reverse transcriptase in order to convert to our mRNA. And so what you really want to consider is what does each group entail? And this is something that I do expect you to know. So yeah, there's a little bit of memorization, but it's more so you guys can understand the concept of how mRNA is made. And so as I was talking about here, you need this minus strand to make mRNA. So if a virus has a minus strand present in its genome, it can directly convert to mRNA. So let's go ahead and go through some of these groups. So in group 1, this is a double-stranded DNA virus. And so basically it's going to have both the positive and negative sense strand. The fact that it has the negative sense strand means that it can directly convert into mRNA. So let's go through here, and who else has a negative sense strand? Group 3 as well. So group 3 is composed of a double-stranded RNA. And so this is also going to have a positive and negative sense strand. But what's important again is this negative strand that's present. If it has this negative strand, it can convert directly into mRNA. And then the last virus that's part of this group that has a negative sense strand are these negative viruses. And so again, because it has this negative strand, it can convert directly to mRNA. Well, then you might ask, well, this double-stranded DNA virus over here has a negative strand, but it takes a couple of steps. Well, again, as I mentioned, there's two groups of viruses, group 6 and 7, that require reverse transcriptase in order to convert mRNA. Why viruses do this, I don't know. I'm sure it's some kind of evolutionary process that occurred, but I don't really know why they do what they do. We just know that they require this enzyme to convert to mRNA so then they can make functional protein. And so with the the double-stranded DNA viruses, although they have this negative sense strand, they require reverse transcriptase to work. But again, as I said with the reverse transcriptase slide, you need an RNA template first. So basically what's going to be happening is this double-stranded DNA is going to convert to a negative sense strand of RNA, and this negative sense strand RNA can be converted to double-stranded DNA. So again, the process from converting RNA to double-stranded DNA is reverse transcription. And so if we look at the other virus that utilizes reverse transcriptase, we see that the positive sense strand of RNA uses it, but because it has an RNA template, it can directly convert to double-stranded DNA. And then from there, mRNA can be generated. Brian, you have a question? Feel free to unmute yourself.
Yeah,
Uh-huh.
I was wondering for the reverse transcription, since it needs to make a strand of RNA first before it makes the other double stranded DNA, wouldn't it make the same DNA as it started with?
What do you mean it makes the same?
So since it would start, it starts with one of the strands it's of its original strands to make the RNA, right? And so then wouldn't that strand then make RNA that is just the complement of the template strand?
Yes.
So when making the actual DNA from it, wouldn't it make just the one that it started with?
Correct. And so I think I might have misspoke. This one's going to be a positive sense strand.
Okay, I see.
Yeah. So sorry. Yeah, because the negative is what's going to be what encodes information. So this will end up becoming a positive sense strand, just like this guy. And then from there, it'll end up reversing everything. And then there's so I'm not going to expect you guys to know the whole mechanism that happens here, but basically, this intermediate step is going to have a combination of a DNA and RNA strand, and then there's a couple of other enzymes that come in and replace those RNA nucleotides with DNA nucleotides. Does that answer your question?
That makes sense, yes.
Okay.
Thank you.
All right, and let's see. Why can't positive strands immediately convert to RNA like group 2? All right, so good question. And so it has to do, going back to this, we need the negative sense strand. So the positive sense strand does not encode for anything, or it's not used for the whole transcription process. So if it doesn't have that negative strand, then it can't make the mRNA. And so if we go through the remaining groups that don't have this negative strand, we see that there's a couple of steps that need to occur first before we can make the mRNA. So for group 2, we have a single-stranded positive sense DNA virus. And so because it doesn't have that negative sense strand, it needs to be synthesized first. Once that's synthesized, then that negative sense strand can be used to make mRNA. And the same concept when it comes to group 4 here. So this positive sense RNA doesn't have the negative sense strand, so it needs to get converted to a negative sense strand of RNA first before that information can be used for mRNA. I hope that makes sense and it answered your question. So I'm going to end up giving you some questions on the exam, and we're going to get some practice in a second. But basically, if I were to give you a viral genome and I tell you whether or not it uses reverse transcriptase or not, I'm going to ask you what are the steps to get to there. And so if you understand this concept of needing a negative sense strand present, then you can go ahead and rationalize what the answers are. And so I have a list of questions you can ask yourself when it comes to trying to rationalize the answers. So when it comes to getting a question as so, you want to ask yourself first, is a viral genome either a double-stranded DNA virus or a negative sense strand virus? And again, because a negative, both of these have a negative sense strand, they can, you follow up with, does it need reverse transcriptase to replicate? And if it's no, basically these two ideas will help you rationalize that it's going to be directly to mRNA. You don't need any other steps to facilitate that mRNA production. So if it does need reverse transcriptase, then of course there's going to be multiple steps that are required, and you're going to be wanting to look for those kinds of answers. And so then if it does need this enzyme, then you have to consider what form is it in. And so if it's in the RNA form, again, reverse transcriptase needs an RNA template in order to convert to double-stranded DNA. Once the double-stranded DNA is made, again, it has that negative sense strand. So then it can get converted to mRNA. So this is going to be group 6 viruses. Now, if it's not in the mRNA form, then basically you need to convert that DNA into RNA first. And then once you have the RNA, you're going to generate reverse transcriptase is going to be used to basically create that double-stranded DNA. And again, because it has that negative sense strand, it can make mRNA from it. And so if the viral genome is either at a double-stranded DNA stage, or if it, sorry, let me back up. If it does not have a double-stranded DNA or negative RNA strand in its genome, then you have to ask yourself, okay, is it a positive sense DNA genome? If yes, it's going to convert to double-stranded DNA first, and then it can make the mRNA. And again, this goes back to because it has that negative sense strand. And then if it doesn't, and if it's RNA instead, basically that RNA is going to get converted to a negative sense of RNA, and then from that, mRNA can be made. So basically, in order for mRNA to be made from for the virus, you need either double-stranded DNA, which is going to encode for that negative sense strand, or a negative RNA strand. All right. So I have questions that are going to be really similar to the exam when it comes to trying to rationalize these answers. I will go ahead and give you guys about 5 to 10 minutes to do these. I'm going to go ahead and put you guys out into breakout groups. And then hopefully you guys can help each other out when it comes to answering some of these. And I'm going to go ahead and stop sharing this for a second, and then let's break you guys all out. All right, I'll see you guys in a second. And if you guys have any questions, feel free to call me into your group. The people that are present, are you guys struggling to get into your breakout group, or are you guys just not present at your computer? I got a question. Are negative and positive strands of mRNA or single-stranded DNA just the complement of each other? Are negative and positive strands of mRNA. So with mRNA, let's see, let's go back here. mRNA is going to have a 5' to 3' direction. So it's only going to be in one direction. So no, it's only going to be one or the other. Hope that made sense. All right, I think everyone's back in here. So let's go ahead and take a poll to see how people are doing and understanding this. Hopefully it wasn't too difficult. Go ahead and we'll wait until the majority of the class has responded. Waiting for about 30% of the class. I'm hoping people remember to write their answers down this time. All right, good enough. I'm going to end the poll now. Share results. So good, you guys did pretty well for the most part. The answer 1 is B. Answer 2 is C. Answer 3 is B. And answer 4 is C. Does anybody want me to go over a particular question? I'll only pick one of them, and then if you guys need additional help, just see me during office hours after this. Let's see, anything? Question 3. All right. Question 3, let's do that one. So what is the next step of a positive sense RNA virus that uses reverse transcription? All right, so the answer, let's just make sure. Yes. So remember how I said before, so the fact that it uses this, actually, before I even say that, what answer did you end up picking for 3, for the people that are asking that they need help? So you guys pick C? All right, so negative RNA. Okay, so if we go back to, let's see. So we have to understand this concept of what is reverse transcription. Okay, so remember, it's the opposite of what transcription is. So transcription is DNA to RNA, and reverse transcription is from RNA to DNA. And so I said here that this virus uses reverse transcriptase. So that means that this, and again, RNA or reverse transcriptase need RNA. So RNA is already present and it's using this enzyme. The reverse transcriptase is going to convert that RNA to double-stranded DNA. So that's why the answer is B. I hope that makes sense. And then if not, go back to group 6 of the Baltimore classification and kind of follow that pathway to kind of rationalize that answer. All right, moving on, as I don't want to go over. So in the end, naming a virus, there's actually no consistency. We're able to group them together based off of the Baltimore classification that I just mentioned, but when it comes to naming them, there actually isn't really any standardized nomenclature. It's really all dependent on who discovered it. And so the only thing that's consistent with the naming of viruses is the suffix viridae. And so all viridae means is family. And it's specifically viral family. So if you ever see viridae at the end of an organism, it's always referring to a family of viruses. And so here I have a couple of examples of the different viridae, but this is not all of them. This is just a couple of highlighted ones. So some viruses are named because of what they look like. So how we just saw in the video earlier, the coronavirus has these spike proteins on them, and they look like a crown. And so corona means crown when translated. So that's why people call it the coronavirus. Sometimes viruses are named because of where they were found. So for example, the Zika virus, that was found in 1946. And it was found amongst gorillas in the Zika forest in Uganda, and that's why it's called Zika virus. And some viruses are named because of the cell type that they infect. So hepatitis is a prime example of this. It infects hepatocytes, and hepatocytes are liver cells. So for the transmission of viruses, all viruses need a living organism in order to reproduce and make more viruses. If it does not have this, it's going to die. And so in order to do this, it needs to find ways of spreading. And viruses have many ways of spreading depending on what family it falls under. So the prime one that we've all been dealing with this last year is respiratory transmission. So prime examples of that are both corona and influenza viruses. There's also fecal to oral transmission. So a prime example is enterovirus. So if you end up defecating some of the virus, you don't wash your hands properly, and then you decide to make yourself a feces sandwich and eat it, then you're going to end up getting infected. Bloodborne transmissions are another way that viruses can spread. Hepatitis B and HIV are good examples of this. And then some viruses can be transmitted sexually, and HIV and Ebola are two other examples of this, and Zika as well. And then it's all dependent on how it's transmitted. And so sometimes it can be transmitted from an animal or an insect, and these are known as vectors. And so the grouping of viruses I do want you guys to know are arboviruses. And so arboviruses are just arthropod-borne viruses that transmit the viruses through an insect vector. And so all a vector is a living organism that can actually transmit the infectious organisms. And so prime examples of these are mites, ticks, fleas, and mosquitoes that are able to do this. But the worst of the worst are the mosquitoes. These are the ones that are responsible for a lot of the diseases that exist in the world. And here I have a picture of the mosquito Aedes aegypti. So there are three main genres of viruses. And so the one that I really want you to pay attention to are these flaviviruses. And these flaviviruses are actually transmitted through this Aedes aegypti mosquito. And so the viruses that I do want you to know from this family are Zika, dengue, and West Nile. Unfortunately, there are no vaccines for this family of viruses, and we only have preventive measures that we can do. So right now it's really insect repellent and reducing standing water, which is a breeding ground for mosquitoes. So a little bit more about flaviviruses. So they are a positive sense RNA virus, and they're always transmitted through mosquitoes. And there's two classes of mosquitoes, but Aedes aegypti is the main one. There's at least 5 different viruses that are found in this family, but the only ones I want you to know are Zika, dengue, and West Nile, as I'm sure you guys have heard of all of them, if not at least one. And basically the reason why I'm putting more of an emphasis on Zika is because there was an outbreak in Latin America in 2015. And so people that were infected experienced some of the symptoms here, such as joint pain and fever and rash, which are common things that any of the flaviviruses will end up causing in an infected individual. But why it was such a big problem is because infected pregnant women where their offspring were developing microcephaly when they were born. And so micro meaning small and cephalo meaning brain. So they were being born with small brains. And we now know, after all the extensive research that's been done, is the reason why this was occurring is basically Zika we now know is one of the few viruses that can cross the blood placental barrier. So the placenta that we're all forming in is meant to protect us, but there's a handful of microorganisms that can cross that and infect the developing fetus. And this infection can lead to a wide range of effects. And so Zika was one of those that could cross this barrier. And so Zika is neurotropic, meaning that it likes to infect neurons. And so when it would get into the developing fetus, the first place it would go was to the developing brain. And so basically the virus would end up replicating in these cells and killing some of them, and basically leading to birth defects. And the prime one is this microcephaly. Depending on how early the infection is in terms of fetal development will determine how dramatic the outcome is. So if a woman is just barely getting pregnant or is in her first trimester and gets infected, chances are the baby is going to develop microcephaly, or more than likely, she's going to end up having a stillborn. And then the later it is, or the closer to development, there's going to be less chances of microcephaly being done, or it'll be less exaggerated. So another group of viruses are viruses that have the ability to cause tumors. And so a prime example of this is the human papillomavirus. And the way it's able to do this is by basically influencing the cell's gene regulatory machinery. And so when it does this, it basically is able to lead to an overgrowth of the cell, which gives this wart-like appearance. And so there are many kinds of HPV that exist, and they're all denoted by some kind of number. So HPV, some number. And the ones that we really care about are HPV 16 and 18, because those are the ones that play a role in developing not only genital warts, but leading to cervical cancer. And so with the development of Gardasil, the HPV vaccine, it basically educates your body to recognize HPV 16 and 19 before it can even really infect a cell. And with the event of this virus, it's actually a very effective virus where we're having 90% efficacy. So get your vaccine if you haven't. And then kind of touching on what I'm going to emphasize more in the next lecture is viruses target specific cells. And what I mean by this is they have the ability to bind to specific receptors. And so not every cell has all of these receptors that, on their cell surface. So depending on what the cell expresses will determine if it's a source of infection for that particular cell. So HIV is a prime example of this, where it basically will basically hijack or utilize these two receptors, CXCR4 and CCR5. And these are expressed on a couple of different immune cells, but the one that is most important to know are the CD4 T cells. And I'm going to get more into these when we're covering immunology, but these are the ones that play a role in adaptive immunity. If you do not have adaptive immunity, you will never clear an infection. And so more importantly, when these cells are getting infected, they're basically being targeted by the immune system to die. And so if you're getting these cells killed by your own immune system, you're going to end up having a weakened immune system. And that's why a lot of HIV/AIDS patients don't really die from the virus itself. It's because they have such a compromised immune system that some other opportunistic infection ends up taking them, such as pneumonia or toxoplasma encephalitis. With COVID or SARS-CoV-2 that we've been experiencing this last year, In order to get into cells, it requires the receptors ACE2 to be expressed on cells. And there's only a handful of cells that express this, and lung tissue is a prime place where there's tons of this receptor. So that's why it's one of the places where the virus is most prolific and creates the most damage. And then poliovirus needs CD155, which is expressed on your skin cells. And from that, it's able to go into the cell. And that's why virus going into that it lives in some pools can infect a person because it gets onto your skin cells, which, you know, we all have on the outside of our bodies. And so once it binds to these receptors, it basically can get into the cell and then begin its whole replication process, which we will talk about in next lecture, highlighting specific viruses. And then here we have basically the treatment and prevention measures we have against viral infections. And so again, once I educate you guys more on the immune system, I will go more in depth in how these therapies work. But basically, just to give you a little bit of a preface of what we have against viruses are really our first line are antivirals. And so in the next lecture, when I talk about how viruses hijack our cells and our replication machinery, basically the antivirals will target specific parts of that pathway to inhibit the virus from replicating. When it comes to vaccines, a typical vaccine will be where you end up killing the virus or weakening it in some way, and then injecting into a person so the body can recognize it. Once the body recognizes it, it can get educated so you can generate a memory response. And so this is how a lot of old vaccines work. But with the coronavirus, or at least the most effective ones that most people are getting, Moderna and Pfizer, that technology is a little bit different, and I will get more into that when the lecture arrives. And then finally, we have these things called interferons. So interferons, so there's alpha, beta, and gamma that are basically how the immune system alarms itself. And once these alarms go off, basically all cells know there's a viral infection going on, protect yourself. And then it'll upregulate or downregulate certain proteins so it can help fight off the infection. And so interferons are a natural part that we express, but companies now are able to synthesize these molecules and actually give them to patients so that they can help kill off viruses and alert your immune system. However, these are quite pricey because of the process when it comes to making these, because there's a whole synthesized processing that is quite costly to make these, so it's not really a cost-effective measure. And so there's a couple of other things that I would like to mention before we end, and that's things that people think of as viruses, but they're not actually viruses. So viroids are an example of this. So they're similar to viruses in the terms that they have a genetic code to them, but they don't have a protein coat around them to protect itself. And these viroids are really important in causing disease in plants. And so if you're a pathogen that infects only plants, it's not going to infect any animal cells or bacteria, and vice versa. All viroids are made up of a single-stranded RNA template, and they're usually passed through seeds or pollen of plants. And what ends up happening with plants, it'll end up stunting its growth in some way. So here we have an example of a tomato plant where uninfected, looks quite healthy, compared to different amounts of viroids that are present within these plants. And so we see the more virus that's present, or sorry, viroids are present, the more stunted the growth is. And then we can see this with a marijuana plant when it comes to its trichrome production. So basically, the more fuzzy it is, the more fats it has that will end up getting high, such as THC. But when a marijuana plant's infected with the viroid, basically there's going to be less production of these trichromes. And then finally, the last topic I want to cover are prions. So prions are also not viruses. And so prions are like viruses, but they are different in the sense that they're not really trying to replicate so much, but they're more causing destruction. And so a prime example of a prion infection is mad cow disease. And so once the prion is in the host, it tends to basically misfold other proteins. And when proteins are not folded properly, then it's going to end up affecting the protein's function. And so prions really like to disrupt the proteins in brain neurons. And so if a person's infected or a cow's infected with the prion, basically it's going to misfold neurons in the brain. This is going to form holes. And then eventually the patient will end up succumbing to the infection. Although the infection is quite rare, it is 100% lethal as we have no treatments or cures for them. So going back to this question of, are viruses alive? And so it really depends on what the definition is of living. And so scientists usually use this, these questions to ask themselves if something is living or not. And so here, the first question, does it have order? Does it have a structure? Yes, viruses do. Can it reproduce? Viruses can reproduce. Once it's inside the cell, it can go to make copies of itself, although it doesn't reproduce the same way that a typical cell would. Does it grow and develop? So what it means by this is we start out as babies and then we become adults and we get bigger as we go. The cells themselves aren't getting bigger, but there's just more of it. When it comes to viruses, viruses will always end up staying around the same size. So they're not able to do this. The other thing scientists use to define a living organism is if it uses some kind of energy process. And so as I was saying at the beginning of the class, they don't have any mitochondria, they don't have any kinds of transporters that exist in or on them, so they're not able to uptake sugars and other macromolecules to perform energy related processes. They are not able to regulate themselves. So like when we get hot or something, our body ends up sweating or some other kind of stimulant that causes our body to react. Viruses do not have any kind of regulatory mechanism. And then this kind of goes into response to a stimulant. So if they're not able to sense anything, then there is going to be no reaction that ends up occurring. And so then the last thing they ask themselves, does it have any kind of evolutionary adaptations? And it does. Viruses end up, when they replicate, they make mutations just like us. And those mutations, like I was mentioning yesterday, have an evolutionary advantage. So I will go and I'll ask you guys these last two questions just so I know you guys have an understanding of this. And then in the process while that's going, I will go ahead and release the sign-in sheet. And if you have any questions, stay after as my office hours will begin. All right, the majority of you guys have answered. Let's go ahead and end this and show you guys. So I'm not keeping you after. Share results. And the answer to both of these was E. The majority of you guys did end up answering. So for number one, basically go back to that diagram I had for the classification for Baltimore, and you'll be able to see what viruses exist. And so again, I do want you to know the different groups that are present. And then the viruses can infect basically all these. I didn't go over fungal viruses, but all types of living organisms have some kind of virus that they can infect. All right, so you guys are dismissed. If you guys have any questions, feel free to stay on.
Hey, Professor, congratulations on getting married next
Oh,
week.
thank you.
You have a good one, man.
Thank you, I shall.
Take care. Bye-bye.
Bye.
Wait, Professor.
Yes.
Wait, is there anything like a double-stranded RNA is special for? Like, why does it exist exactly?
I don't know the answer to that. I don't know why viruses form. If I had to guess, I'm sure a lot of the viruses are related to one another if you look at it genetically. But as the more times viruses replicate, they're actually very error prone. Meaning that once the polymerases are adding on and replicating their genome, they make a lot of mutations in it. And so it's possible that there's something that went wrong in that process, but the genetic material was still able to survive, and then that survival allowed for it to branch into something else. But I really have no idea why the viruses exist.
Thank you.
Mhm. So the people that are left, are you guys not present at your computers again, or do you have questions? So I understand that you were here during lecture and were watching, so you didn't miss anything, but that's not the point of coming to lecture. It's part of it, but the point is so you can participate. These points are for participation, not for simply coming to lecture. Is that understood? So I'm going to assume the two people that are still here are not at their computers and weren't paying attention.